Good evening, Anna. Good evening.
Good evening, sir.
Good evening, Anna. Any doubt in previous chapter, Vijay? Do you have any
No,
doubt?
sir, no doubt, sir.
Suppose if you have any doubt, you call me, okay? You ask me, okay? No need to worry. Be free, okay?
Yes, sir.
Mm-hmm.
Sir, you can start now, sir.
Yes, ma'am. Thank you. Thank you so much. So, good evening, my dear students. Okay, today what we are going to discuss about, before that, already we saw it. What are the pollination? What is meant by pollination? What are the types of pollination? And what are the factors involved in the pollination process? All those things already in our board. The pollen grain, transfer of pollen grain from the anther to stigma is called as, what is that? Pollination. Already we discussed. Okay. See, today what we are going to discuss, the artificial fertilization. Artificial. Before that we studied, they all are comes under the heading is known as natural, what is that? Pollination. Fertilization also called as, another name is called as pollination. Okay. So artificial means man-made. Okay. What is the main reason for the artificial fertilization? We can easily cross with any plant, same species, but any plant we can cross. As a result, what will happen? You can get a improved plant. Okay. Take an example. Two species. One is the food quality or whatever it may be, whether it may be tall or short or straight or weight. According to their weight, you have to select the corresponding flowers. Okay. And also particularly tell us the anther of the particular superior quality and characters. This is very, very important. Okay. So how they are doing the artificial fertilizations? Those things we are going to discuss. See here, artificial fertilization. It is also one of the type of cross pollination performance by man for crop improvement achieved by two processes. That how they are achieving the artificial fertilization means, first one is called as emasculation. I have to note it down. Emasculations. Okay. This is also already so many times they were asked. Okay, what is my emasculations? It is nothing but the removal of anther from the flower when in bud condition. That is very, very important. During the bud condition only, we remove the anther of a bisexual flower. About male as well as a female sex organs, otherwise called as stigma and style and ovary is a female flower, and anther, otherwise called as stamen, is a male reproductive organs. Both are present in a single flower is called as bisexual flower. Okay. See here, during the bud condition, both are present in it, but it is not mature. Note it down. That point is very, very important. Why they are removing during the bud condition, because it is not well happening. We have to remove the anther. That is very, very important. Before the anther dehiscence, using a pair of forceps. You have to use how they will remove the anther means with the help of the forceps. Okay. During the bud condition in bisexual flowers, they may ask the questions. Okay, in this area, it's very, very important. After the removal of anther, what you have to do? Any one of the insects, any one of the pollinator, take an examples, abiotic factor as well as the biotic factors. Okay, otherwise called the biological factors. Okay, then they, what is that? Take the anther from the another plant, and also it will be easily fixed with the, it is easily fertilized with the this what is the removed anther area. No. So what happened as a result? It can be cross pollination is possible. Okay. So you will not get the good quality. That is very, very important thing. That's why what we are telling, emasculation, that is removal of anther from the flower in bud condition of a bisexual flower before the anther dehiscence. Before dehiscence, you must remove the anther. Suppose if it is dehiscence, what happen? It may be spread the pollen grain where it may be reach the, what is that? Stigma. As a result, the formation will be, that means zygote formation, it will be possible. So before dehiscence, using a pair of forceps, we remove it. Then what you have to do after that, removal of the anther, we have to bagging, otherwise called as packing. Okay, covering of the emasculated flower with a bag of suitable size to protect them from contamination with unwanted pollen. That is the reasons why we are packing this one after the removal. So that is called as artificial. Okay, that is called as, it is a man-made, So man-made means first removal of the anther, otherwise called as emasculation. After that, we have to packing the, what is the flower with the suitable size of protected polythene bag. Why we are covering the emasculated flower? Due to which one? It may be protected from the contaminated with unwanted pollens. That is very, very important. Okay. Okay. Then, if flower is unisexual, suppose if it is flower is unisexual, emasculation is not needed. In our unisexual means the male flower is in separate plant and female flower is separate plant. No need emasculations. This is also another point. Okay, then flower bud backed, and when the stigma becomes receptive, pollination is done using the desired pollen and the flower is rebacked. Once again, emasculation done in a bisexual flower. After that, you are going to packing. When it undergoes the maturation, which one? The female, that means ovary or stigma tail ovary, that is when it undergoes the maturation process. After the maturation of the female, what is that? Reproductive part, we can pollinate with the receptive and also desired quality and desired pollen grains from the any one of the same species. For example, suppose mango is one of the species, sapota, take an example, apple, orange, whatever it may be. Okay, and also after the unison of the different species, the cross pollination is also done. After that, we have to reback. Once again, you have to tie the, what is the flower with the polythene paper. Okay, see the next. So this is called as a emasculation. This is the diagram. This is the forceps. Already you know about it. Okay. So removal of anther in bud condition. This is a bud. Listen here. This is the bud condition. Here the center part, what they are having, this is a female reproductive part. This is called as stigma. This one is called as tail. This one is the part of ovary. So the near there is a small protrude-like structure, isn't it? This is called as, what is that? Immature anther. Before dehiscence, mature anther means what happened? It may be growing the similar to this level. Okay, but here immature anther is there. Before dehiscence, we have to remove this one. These are all are must remove it. Then only after the removal, what happened now? You have to pack with the polythene bag in corresponding, which one polythene bag. Okay, it just here you saw this diagram. What you are seeing, see that this is the female reproductive part, male part is removed. Think. Okay, so go to the next one. See, this is the way, like this. After the removal of the which one? Male reproductive part, it look like this. This is only the presence of female reproductive part only. After that, you have to cover it. How? See. So you have to cover it. This is another plant, rose, I think. Okay, you cover it like this, and also you have to tie here. Okay, tie it. Then suppose some of the pollinators, they may be enter inside like this. Excuse me. Enter inside. So as a result, the cross pollination, what we are desiring, and that quality will be completely contaminated. So that's why, what is that? This is a method of covering. After that, you have to tie it. Okay, properly. Okay. And after that, what happened? Already you know very about. When the male as well as the female gamete united, what happened? Automatically the fertilization takes place. Okay, suppose if it is occurs in the angiosperms. Suppose if it is occurs in the angiosperm. Mostly you know very about most of the angiosperm double fertilization is possible. That is very, very, very important. Okay, but now what happened? The anther is removed. Okay, at the result, what happened? Another cross pollen is also we done it. As a result, also it tie with the corresponding polythene pack. Inside what happened? The fertilization takes place. As a result, double fertilization. How already in our what is meant by ovary structures? What are the cells present inside? Okay, what are the nucleus? How many cells are there? Already we saw it, isn't it? How the pollen tubes enter? There are three ways. Is there? Poro and porozoic. Sorry, I mean, poro, and then another one is called as salazo, and then third one is called as what is that? Mesozoic. Sorry, meso. Okay, these are the possibilities. Okay, already we studied that things is also. Now we are going to study the remaining things that is called as double fertilization. After the covering, inside what happened? Fertilization takes place. What fertilization? Double fertilization. Why? Because it's a angiosperm. Okay. See, the fertilization takes place twice at a time. An angiosperm is called as double fertilization. Why it is called as double fertilization? What is meant by fertilization? Fertilization, nothing but the fusion of male as well as female gamete within the female ovary is called as fertilization. Double fertilization means double fertilization takes place. That means double male gamete united with the double, what is that? Female gametes. That is called double fertilization. Where it occurs? Angiosperm, it occurs. Which one is the examples? Typical angiosperm is an example. Example name, capsula. Okay. See here. The double fertilization was first discovered by the name of the scientist is called as Navaskin. This is also so many times they were asked. The double fertilization was observed by or discovered by the name of the scientist. This called as Navaskin. Take a notes. It's very, very better for your future references. Okay, which one is I am conveying committed the words, which one is important for the NEET? You just note it down and also you write it down. Okay, it's very, very better. Take a notes, that is also very, very important. It's just put a note also. Okay, double fertilization was discovered by Navaskin in Lilium and Fritillaria plant. You note it down, Lilium and Fritillaria plant. Okay, so the next, the double fertilization and dribble fusion. Already know very about what is by dribble fusions. What is by double fertilizations? That things already known. Is the specific or universal characteristics of angiosperm. This is already we discussed, isn't it? Because most of the angiosperm, the double fertilization is possible. Double fertilization, how many fusions are there? Triple fusions, but two, what is that? Fertilization takes place. That's why it is called as double fertilization and triple fusion is there. Okay. So there are 5 nuclei and 3 gametes participate in the double fertilization. This is also a very, very important. How many nuclei present during the double fertilization? How many gametes participate in the double fertilization? It may be given in the number aspects. Number means in our 5 and 3, 5 nuclei and 3 gametes, like that they may give. Okay, so you have to point out these things also. There are 5 nuclei and 3 gametes participate in the double fertilizations. Okay, after entering the one of the synergids, after the what is that? The pollen tubes, when it enter into the micropyle through the which one filiform apparatus, when it reaches the synergids, the pollen tubes releases 2 male gamete into the cytoplasm of the which one Okay, see that cytoplasm of the which one female synergetics. That means otherwise called as female reproductive part. As a result, what happened inside? 2 male gametes enter. See that this is the entire diagram. So this is the already in our but this is called as what is that? Ovary. How many cells present in it? There are 8 cells is there. Already we discussed these things. Here there is a 3 cell is there. Which one is a orange color or yellow color? Okay, this is considered as a X cell, and 2 remaining is that this is called as a lateral side. This is also a what is a synergetic. What about the central? This one is called as antipodal cells. Say this is the pollen tubes. Just imagine this is the pollen tubes. It's also produce the two gametes. This one is called as what is that? Generative cells. This one is called as tube cells. When it enter before that, what happened? It automatically, mitotically divided into two. Okay. When it reaches the, which one, microcells, it releases the two, what is that? Male gamete inside. One gamete, one male gamete fused with the egg cell here. See here, this one is the egg cell. This one is called as a, what is that? Male, otherwise called as male gamete. So one fusion takes place. During this result, what happened? The center nucleus also fused. So here the fertilization takes place. Here the fusion takes place. This is also a one of the fusion. So both are called as a, what is that? One fertilization possible, and second one is fusion possible. But two fusion is also done here. And the next one is that another male nuclei also connected with the this one. Okay, the central nucleus. It is also converted to primary endosperm cells. This one is called as, what is that? Egg cells, when it's combined with the male gamete, form as a embryo. This is called as embryo. This is called as, what is that? Primary endosperm cell. The primary endosperm cell converted into endosperm and provide the nutrition for the, which one, embryo. We'll see the slides. You can easily understandable. See here, the same diagram has given here. Okay, same diagram and different angles they have given. See here, this is also same. So tube cells and generative cells is also present in it. Okay, it enter inside the poronic microsporopyle. So when it enters, what happened? There are three stages there. Here also two cell. Okay, how many nucleus is there? See, the two nucleus also go join with the corresponding male and female, isn't it? So here two nucleus released by the pollen from the pollen tube. So it reaches the egg cell, and another one is it reaches the, which one, the center cells. Okay, so this one is considered converted into what? We'll go to the next slide. You can easily understand. Okay, see. So these are the type of what is that? That fallopian tubes. The tubes enter inside the ovary or ovules. Sorry, ovule. Okay, progamy. See here, this is called as progamy. The pollen tubes enter through the way microphyle is called as progamy. Already we discussed. Chalosogamy, it enters the chalosa part. This is the opposite part. This is the sporogamy part, a microphyle part. Opposite, there is a part is called as, what is that? Chalosa. The structure of ovule, when you are going to study, you study these things also, isn't it? Okay. Chalosogamy, mean enter into the chalosa part, and also again it enter into the microphyles. This is the penetration way. This is the penetration. The inside enter, and also it maybe reaches the microphyles. Once it reaches the microphyle, already same process occur inside. And another one is called as, this is called as mesophyll. It is the porophyle area, porophyll microphyll end. This one is called as chalosa end. In between it enters the, that's why it is called as mesogamy. Progamy, chalosogamy, and mesogamy. Meso means middle. Chalosa means chalosa part, but it enters through the way of microphyll. Progamy is also directly to enter into the porogamy. Enter through the way of, which these are the three ways. Okay, after reached, what happened inside? See, the first one, syngamy, is possible. Already I told you, one of the male gamete fused with the egg cell to form a diploid zygote is called as syngamy. Already told you, any one of the male gamete fused with any one of the, what is that? Zygote. That process is collectively called, what is that? Syngamy. Then what happened? This is the first process occur. That means first fertilization possible. Then two polar nuclei, where it is of the central cell. Once again, see that here the one fusion takes place, isn't it? One male gamete fused with the one egg cell. So fusion takes place. As a result, fertilization is also possible. Upon one fertilization is finished. Two polar nuclei of the central cell fused to form a diploid secondary nucleus. This is called as another fusion. The first fusion, this one is called a second fusion, but it's not a fertilization. Listen here. This is not a fertilization. This is only for the fusion of two nuclei. Which one? Two polar nuclei united together and form as a single nuclei. Okay. See here. The third one is called as triple. The second male gamete fused with the secondary nucleus to form a triploid primary endosperm nucleus. That is simply called as PEN. This is already so many times they were asked. PEN, primary endosperm nucleus. Okay, expansion type of questions. Likewise, PCR, polymerase chain reactions. Okay, so DMLT, diploma in medical lab technician. Likewise, they were asked. What about ELISA? Enzyme linked immunosorbent assay. What is meant by HIV? Human immunovirus. Likewise, yeah, good, Kana.
Sir, this ELISA
So
is a
this,
diagnosis for HIV one, sir.
Yeah. Whether the person suffering from AIDS or not, it is not a confirmatory test. This is only for the person suffered by AIDS or not, like that. Suppose when you are moving to the Western blot method, you know, that method is confirm the person suffered by AIDS, like that. Okay, so these are the things. Okay, go to the next one. So these are the overall, what is that? Events. The first fusion of two polar nuclei, n plus n, you will get 2n. Okay. Second fusion, a male gamete fused produced of the two polar nuclei, that is called a 3n stage, fuse with the fusions. This is also another. Third fusion of male gamete with the egg cell. So this is the first reaction. Okay, third reaction is the first one, and second one is what happened, you know. This is the second one. This is the third one. So likewise, okay. So this is the way, but so here I have listed what are the first fusion, second fusion, third fusion. How many gametes is that? 2n plus a 3n. Here also then total 5. Okay. See here. So since two types of fusion syngamy and the triple fusion takes place in the embryo sac, the phenomenon is termed as a double fertilization. Already you know about. Okay. The two type of fusion of syngamy and the triple fusion takes place in the embryo sac. The phenomenon is termed as a double fertilization. Okay. The central cell, after the initiation of the central cell, what happened? After the triple fusion completely finished, what are the changes occur inside? Okay, see that it becomes a primary endosperm cell. That's why I told you. The triple fusion center cell, you know, it developed into which one, you know, primary endosperm cell. The primary endosperm cells and develop into the endosperm. That's why I told you. The center, what is that? Which one is? The in center, there is a nucleus, isn't it? This one. Okay, this one, see the previous one, you can easily understand it. So this one, see here. So here the one male gamete united with the egg cells. This is a one fusion finish. At the time, what happened? The center nucleus also united. Okay, n plus n. Another male gamete. See here, the red color is a male gamete, isn't it? It's also transferred. The arrow is given here in light manner. Okay, just imagine here transfer. So this one is called PEC, primary endosperm cell. The primary endosperm cell finally develop into endosperm. Already told you, the endosperm provide nutrition for the egg cells, otherwise called as embryo. So what happened here? The first fusion is the important one. The first fusion is what is that? It is converted into, it is developed into which one, you know? What is that? Embryo. Okay, that is very, 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 very important. Okay, the embryo is this is the double fertilization. Okay. So after that, see that. Okay. The zygote develop into an embryo. I told you, you know, first fusion takes place inside. Male gamete, female gamete united. As a result, zygote is formed. After that zygote formations, these two events also continuously, what is that? Happened. After that, the overall reaction completely finished. That means fertilization finished it. Okay. So finally, what happened? The zygote developed into embryo. The central cell converted to primary endosperm. So total of what are how many fusions are there? There are 3 fusions already finished. See. So this is the total things. Final, what happened? Degeneration of the antibody. Antibody cell completely disappears. This one is called a center. There is a primary endosperm nucleus. This is the main important things. Primary endosperm cells. This one is zygote. The zygote converted into what? The zygote converted into what? Embryo. Primary endosperm nucleus converted to what? Endosperm. What about the synergetic cell? Synergetic cell also degenerated. Antipodal cell also degenerated. How many cells present inside? Only the center cell as well as the zygote only present inside of the embryo sac, or otherwise called as ovule or ovary. Okay, this is very, very important. Final, how many cells are present? Only after the triple fission finish it, there are 2 cells only present in it. One is called as primary endosperm nucleus cells, PEC. Another one is zygote. Okay, the zygote develop into what is that? Embryo. Then embryo, how it will be? Grow as a cotyledon. That things we are going to study one by one. Are you clear this area, students?
Yes, sir.
No. Any doubt means please ask me. Suppose if you have any doubt, will you please ask me? I will clarify it. Which portion is very difficult? This is very, very important. So many times they were asked in this area. That's why I am discussing. Oh, this is a overall event. See, listen here. This is a flowers. Already you know about. The male flower is the reason. These are the male flower. How will be produced? The which one up to the level of tube nucleus. See here, the single cell convert to mitotically 2 into 4. 4 cell is produced. From these 3 cells are completely degenerated. One is enter into the what is the further production of the pollen grains. This is a pollen grain. Mature pollen grain contains only 1 cell. The 1 pollen grains, 1 nucleus is there. It's divided into 2. Already know there are 2 types of nucleuses there. Okay, mitotically divided into 2. So you will get one is called a what is that? Tube nucleus. Another one is called a generative nucleus. The generative nucleus divided into 2 males gametes. So total 3 cell stages there. The 3 cell stages, what is that? The inner side exine entine is the pollen grain. Produce the 3. What is the nucleus? It once it reaches the female. Again, it is up to this level. Okay, the first one is first one. It is considered as a male reproductive up to the level of the pollen tube formation. Next one, same is that. Already you know about the single archisporial cells. Okay, so there is a single cell is there. Single nucleus, single cell. Divisions takes place. How many cells are there? 4 cells. 3 cells are completely degenerated. Only 1 cell is there. The 1 cell divided into 2. They move towards the pole. Okay. After it reaches the pole, what happens? A mitotic divide twice. You will get 2 and 4 and 8 cells. After the formation of 8 cells, the 1 cell move to the center, and also the antipodal cell also move to the center. This is the stage. So 3 is the top, and center there is 2, and the last is the 3. Okay, total 8 cells. This is called as mature ovary. This is called as mature, what is that? Pollen grains. So after the maturation process, that is called as microsporogenesis and megasporogenesis. What happened here? The male gamete united with the female gamete through the way of pollen tubes after the fertilization. This one. Okay. These are all before fertilization. This one is called as at the time of fertilization. These are all called as after the fertilizations. This is very, very important. After the fertilization, when it reaches the pollen tubes, so what is the double fertilization also possible? Triple fission takes place. What happened? Only the 2 cell stage. Before that we studied, 2 cell is the, again it undergoes the divisions. It produce the, what is that? Embryo. The embryo, it contains, what are the parts is there? Embryonical axis as well as a radicle and plumule. The radicle and plumules, what happen, automatically or correspondent parts, they will be produced respectively. Okay, how it will be produced? Radicle produce, what is that? Root system. Plumules produce, what is that? Shoot system. These are the 2 cotyledons. Okay, they provide nutrition for the, what is that? Embryo. Finally, it will reach us the, what is the entire plant. Again, the cycle will be repeated again and again. Okay, because this is the well-developed plant. That is called as angiosperm. The initial stage, this is called as plantlets. This is called as cotyledon. This is embryo. Embryo is grow into a plant. Finally, it reaches the reproductive part. Again, its the cycle will be continue many times. This is called as what is that? Polycarpic plants. Okay. So here, any doubt is there, my dear students? This is the complete event up to this level. What we have already studied. Okay, complete event of the lesson given in a single slide. Okay, male gamete, female gamete, how it will be produced, double fertilization. After that, embryo, how it will be grow, what is my plumulent radicle, entire plant, again juvenile stage 2 at reproductive stage, corresponding male and female gamete production takes place. Again and again, it will be continued. Okay, see the work to the next one. So, I think the lesson second part finish it. We'll pass on to the next one, third one. Okay, see that. Do you have any doubt, my dear student, here?
No, sir.
Right, boy. Listen here. Last time you asked the one question doubt, isn't it? That one is called as Euclina, isn't it?
Yes, sir.
Yes, that one is correct. Why I am telling because it's a little confusion. It may take why both whether paramecium as well as the Euclina both are structure will be similar, isn't it?
Yes, sir.
Yeah, only one difference is there. Will you please explain it? Which one is the difference between these two? Which is
So
the
Euclina
different?
has flagellum, paramecium doesn't have flagellum.
One more is there. Euclina it has one more is there. Will you please?
So Euclina has some structures that are present in plants alone, only chloroplasts.
No, yeah, very good. Correct. Exactly correct. The Euclina, it has a chlorophyll molecule. It's not only that autotrophic, and also it has a heterotrophic nature also. It is one of the moving animal is called as what is that? Euclina. Okay. So that one is correct. No need to worry about that one because it is a simple things. Okay. But transfer, what is that? Fission takes place only in the, which one Euclina only. Because paramecium only the transfer section is possible. Okay, you got it? You clear that one?
Yes, sir.
Okay, no need to worry because it's a simple things only. Because the structure will be similar. That's why you may what is that? Sometimes it may be a doubtful one. That's why I'm clear it. Okay, go to the next one. Okay. Okay. Carry on. See the next one after the fertilization, what we are going to enter in the post-fertilization. Okay, post-fertilization, what happened? What are the changes, morphological structure changes which occurs within the ovary? Post-fertilization, all events that occur in the flower after double fertilization is called as post. I told you after the double fertilization finish, okay, what are the changes occur? That is called as post-fertilization. What are the major event occurs? See, these are the events. There are 4 events have been occurred inside the what is the ovary. After that, see, first one is development of endosperm. Already told you endosperm. Which one is the endosperm conversion? Which nucleus is converted into endosperm? Central nucleus, isn't it? Polar nucleus. Okay. Development of embryo. Embryo is nothing but the egg and the sperm. Otherwise called as a egg nucleus of and also anther nucleus. Otherwise called as a pollen grain. Sorry, pollen grain. Okay. Maturation of ovule into seeds. Normally, maturation of ovule into seeds and maturation of ovary into fruit. These are the events which occur inside the what is that? Ovary. Okay. Post fertilization. After the double fertilization, these are the event takes place. One is endosperm formation, embryo formation, ovule converted into seed, and ovary converted into fruits. Okay, see that? See. Endosperm. The development of endosperm takes place before the embryo development. Once again, they may ask the questions. Okay, the endosperm and the embryo, which one is taking first? Like that they ask, no, endosperm taking place before the embryo development. Embryo development before, which one happen, endosperm development taking place. This is very, very important. Then how it will be? Primary endosperms divided repeatedly to form a triploid endosperms. Okay, this is the very, very endosperm cell divides repeatedly again and again, the mitotically. What happened, until the triploid nature. That means N divided into 3 N stages. Cells are filled with the reserve food materials and are used for the nutrition of the developing embryo. So after the formation of endosperm at 3 N stage, what is the reserve food material filled, okay, and develop for the, which one, it provide the nutrition for the embryo. Okay, then what happened? The PN undergoes successive nuclear division to gives rise to a free nuclei. This is called as free nuclear endosperm. After the PN, what happened? It what is that? Gives rise to a free nuclei. They may ask the questions: which one it gives? Which one Which one it gives? Free nuclei. Like that they may ask. Which one? PN. Okay, undergo the successive nuclear division to gives rise to a given rise to a free nuclei. This is called as free nucleus endosperms. See that? So this is the total endosperm types. Already told you what is my endosperm. Endosperm is nothing but the nutritive tissues. Once again, repeat for you. What are the types of endosperm? Here, what we are going to discuss about here. What is my endosperm? What are the type of endosperm here? What is the main functions of the endosperm? Endosperm is nothing but it is a nutritive tissues for the embryo. Okay, then it is absent in Orchidaceae, in our fruits, Podotemaceae, and also Trapeaceae. These are the 3 families. Which one is absent? Endosperm is completely absent. Okay, when you are going to study non-albuminous endosperm, that means presence of endosperm. Sometimes it may be absence of endosperm. Then how the plant will get the food or nutrition? Okay, that things we are going to discuss later. Then, so oily endosperm. Oily endosperm means now we are consuming the coconut. It is one of the endosperm. Okay, when you are consuming the tender coconut. Okay, tender coconut. When you are consuming, when you are drinking the tender coconut, what will be the? What it will be? Boys or girls? Anybody else? When you are drinking tender coconut, isn't it? It is also one of an endosperm. Which endosperm that one?
Yes, sir.
What is that?
Sir, it is sir, that white thing inside that coconut,
That is
sir.
okay, bye. I'm telling you about that liquid, liquid part. Tender
Yes, sir.
coconut. Tamil we may tell as elani. Okay. What is that? So it is nothing but that liquid endosperm. Okay, got it. So it is also
Yes, sir.
yeah, it is also a very, very important one. A liquid endosperm. Right. Here the oily endosperm means what is that? Coconut, castor, and cotton. These are the important things. That means oil substances present in the endosperm. They are all concerned which type of plastids. Can you explain it? This one, which type of plastids? I'm asking. There are 3 type of plastids, isn't it? Will you please? What are they? Do you know? Okay, I will tell you. Chromoplast, chloroplast, and elaioplast. The elaioplast are colorless, mostly present in the oil seeds. That's why I'm telling you the oil endosperm is found in the coconut, castor, cotton. These are all look like a white and pale yellow or white color. Okay, the oil substances. Okay, present in it. That means food material is stored in the form of oil. That's why it is called as oil endosperm. We'll get the coconut oil, castor oil, cotton seed oil also there. Okay, so these are all. The cotton seed also comes under this one of the very, very important thing. You know about the fiber. Okay, we are wearing the cloth, isn't it? That cloth also made up of which one, which type of fibers? It is nothing but the secondary fibers, past fibers. Okay, they may ask the questions. This is also surface fibers. Cotton is one of the surface fibers. You note it down. These questions are already asked too many times. Okay. What is the next one? Proteinaceous is endosperm. Proteinaceous. It is made up of protein, mostly found in the pulses and cereals, particularly cereals. Take an example. Is found in the cereals. Alluron layer. Already told you. Alluron layer is nothing but the any one of the what is that? Cereals. Cereals. Okay. There is a outermost layer is covered with a layer is called as alluron layer. It is nothing but the protein-rich layer. Okay. And starchy endosperm is found in the cereals too. Same. Okay. Rice and wheat and so on so. Hemicellulose endosperm is found in the date palm and also coffee. Hemicellulose endosperm. A cellulose is one of the, what is that? Structural polysaccharides. Okay. These things no need, but you note it down. And cellulose endosperm is found in the ivory palm. Ivory palm. Palmaceae family comes under the reason. Palmaceae family. The endosperm is the edible part is what is that? What is that? Cellulose endosperm. Double endosperm is found in coconut. Why they are calling double endosperm? Will you please? The white color we are consuming, that is called as what is that? Solid endosperm. Inner there is a small amount of water present in it. That is called as liquid endosperm. So both are present in the single. What is that? Fruit is called as double endosperms. Okay. Okay. Go to the next one. Ruminant. What is the meaning of ruminant? Chewed endosperm is found in the passiflora and also Miritica, Erica. Erica is nothing but what is the that Tamil we may call as Paku. Okay, so this Paku cheding so long. Okay, ruminate, chewed endosperm. This is only used for what is that? Sieving process. Okay, just like a sieving gum. Just take an example, sir. Bubble gum like that. We can. Okay, just think that one, achieved one. Hard endosperm. It is very, very important. They may ask the question. Hard endosperm is found in the date palms, Erica, and also Phytelepus. Phytelepus. Hard endosperm found in palms, and also Erica, and also Phytelepus. Mosaic endosperm. This is also a different. Mosaic endosperm is found in maize. These are the type of endosperm. Endosperm. What is the meaning of endosperm? How it will be formed? Okay. What is the main use of that one? Okay. So what are the type of endosperm here? Already we discussed. Okay. Here you must note it down. Which one? The oil, proteinaceous, starchy. Which one is double endosperm and also ruminate and hard endosperm. And last one is called as mosaic endosperm. Mosaic maize. Mosaic maize. Like that it is easy. Spelling is also pronunciation will be different. But, see that. Okay. So then, what are the polyploidy of endosperm? Before we studied what are the type of endosperm according to their nature. And ploidy of endosperm, it's mostly depends upon their number of what is that component endosperms. Suppose if it is only one is present in it, there is mostly in gymnosperm. You know what about gymnosperm? It is also similar to that of the angiosperm. It is also flowering, but it is naked one. Okay, that condition is called as only one end stage. That is called as what is that? Single end stage. What about 2N? Enothera. Enothera is a diploid stage. And triploid in the angiosperms. Most of the angiosperm triploid nature. Endosperm. In our already we studied that before double fertilization, the center there is a tube nucleus already fused together, and also another male nucleus also united together. As a result, you will get it 3N stage. So most of the angiosperm 3N stage only. And 5N, sometimes some of the plant they are having a 5N stage. Panania. Pene, yeah, and also Blumbago, Fritillaria. These are the three families. BPF. Okay. And there's 9N stage is also there. That is called as Peperomia. That is called as Peperomia. 9N stage. This is a very, very, very complicated and also different. See here, how many N stages are? 24,576 N in arum maculatum. This is a exceptional, extraordinary endosperm nature, in plied nature. Okay, see the diagram, see the plant. This is the plant. This is called as aurum maculum. Okay, how many? 24, 5, and 76. See, here remaining, what happened? You know, 3 is absent. In between 2, 3, 4, 5, 6, 7, like that. Imagine it is very simple. 245 and 76. Note it down. Okay, so this is an exceptional case. Okay, so this is the plant. Just to see the structures. This is contain how many? 24, 5, 76. See. Subsequently, cell wall formation takes place. After the endosperm formation, what happened? You know, subsequently the cell wall formation also possible. Place and became a cellular endosperm. The coconut water is free nuclear. Already we discussed the free nuclear endosperm or liquid endosperm, and the white kernel is the cellular endosperm. You know very well about. Already told you there are 2 types of endosperm present in it. Which one we are consuming? That is called a cellular endosperm. Which one we are drinking? Okay, that is in the form of tender coconut. That is called as liquid endosperm. These 2 are present inside. Which one is free one? You know, that is called as free nuclear endosperm, free nuclear endosperm or liquid endosperm. This one is a free one, and the white canal is the cellular endosperm. It is not a free. Okay, the endosperm may be consumed completely during embryo development. Just to note it down, this is very, very important. When the embryo development takes place, all the nutrition of the endosperm by utilized by the embryo, or what happened, you know, or it may be consumed during germination or seeds. Suppose when the seed germination takes place, complete endosperm utilized by which one? Embryo or what is that? Seed. When suppose if it is seed utilized, you know, what happened, you know, it may be consumed during germination. Suppose the embryo utilized 50%. Imagine remaining 50%. What happened? You know, seed germination. During the seed germination, complete endosperm may be consumed completely. This is very, very important. Which one used first? Embryo. Remaining used by seed germinations. This is also a very important thing. See that? This is the way. Next one is called as embryo. First endosperm formation, finish it. After the what are the type of endosperm? According to the plied, how they are divided, all those things will study. Given examples, that things also already studied. Finally, liquid endosperm and also solid endosperm. Give an example. Suppose if it is present double endosperm, which one is an example? Coconut is an example. Coconut, the yellow, white color part is called as what is that? Cellular endosperm. And then inside there is a liquid is that is called as liquid endosperm. Okay. The endosperm completely utilized which one? When it embryo develop, otherwise it utilized by which one, you know? See, germination. Okay. Next, embryo. How the embryo formation takes place? After the formation of zygote. A zygote formed and placed at the micropylar end of the embryo sac. Already you know about. The ovule is there, the lower part, there is a micropyle is there. It is the way to entry of the pollen tubes. That place only, which one in present in embryo present in it. After that, zygote start its development only after some amount of endosperm formed. Okay, some endosperm formation takes place. After that, zygote start to development of the only after. Already told you. First, which one is formed? Endosperm formed. Then only embryo formation takes place. So, some amount of endosperm formed after the formation of endosperm, the zygote start to develop. See that embryo consists of embryonical axis converted to cotyledon. The cotyledon contains 2 parts. One is called as plumule, another one is called as radicle. Plumule gives rise to shoot system, radicle gives rise to root system, because the first letter is just noted on root, radicle. Plumules, what is that? Shoot system. Like that you have to put. So embryo consists of this is already they may ask the questions. Which one is the correct order? Like that they may ask the embryonical axis, plumule, cotyledon, radicle. They may be mixed there. You have to tell which one embryonical axis, cotyledon, and plumules, and then radicle. You note it down. This is also a very, very important thing. See, this is the what is that? Mature embryo. This is called as embryonical axis. Okay. The embryonical axis, the lower part, which one you know? That is the radicle. This one is called as cotyledon. Okay, these two are cotyledon. The center there is a what is that? Plumule. From this only the shoot system originated. From this area only the root system is originated. Okay, these are the what? The embryonical axis. Then, how the embryo development takes place? Zygote, proembryo, globular embryo, and heart-shaped embryo. Finally, it converted to embryo, mature embryo. This is also so many times they were asked in the which one in a correct order. Zygote, proembryo, globular embryo, heart-shaped embryo, then mature embryo. See here, zygote, first one, divided by mitosis into suspensor and embryo cells. The suspensor cell forms a globular basal cell which remain embedded in the endosperm, a multicellular suspensor bearing the embryo. Here, zygote divided into two cells, suspensor and embryo. Suspensor is nothing but the holding of the embryo. So lower part is called a suspensor cell. It's nothing but the stalk-like structure. We'll study the diagram. Okay, when you are going to see the slide, next slide, it will be better understanding. The above part is called as embryo cells. Okay. Then what happens here? The suspensor cell forms a globular basal cells which remaining embedded in the endosperm, a multicellular suspensor bearing the embryo. See, globular embryo becomes heart-shaped. This is also already we know about. Okay, we'll see. And then the mature embryo with what is that? Radicle, plumules, and cotyledon. The mature embryo contain what are the three parts? You know, radicle, plumules, and cotyledons. That is the main one. The mature embryo already we saw it, isn't it? Before slides we saw it. That things is also given here in the correct order. Okay, see the next slide. See, this is the structures. So this one is called as after the what is that? Fertilization, double fertilization. Okay, triple fission finished. You know, these are all are disappear. These are all are disappear. Only the two cells are present in it, isn't it? See, this one is called a zygote. This zygote is here. The zygote undergo the cell divisions. Okay. As a result, what happened? Two cell stage. Two cell stage. This is also, these are the two cell stage. The upper part is called as basic cells, otherwise basal cell, otherwise called as suspensor cells. Okay. See that after that, what happened? You know, this is the what is that? Main important things. So these are the suspensor cells. It's suspensor cells present. These two parts present in it. This is called as globular embryo. They may ask the question, diagrammatical question. They may ask the questions. Okay, the given X. Suppose here they may mark this X or A like that. They have given here. This part is what? They may ask the question. And also which one? This part is what? Like that. They may ask the question. So you have to clear it. Okay. This cell is called as suspensor cells. The suspensor divided into like this. The lower part globular embryo converted to heart-shaped, well-developed. That is simply called as heart-shaped embryo. The heart-shaped embryo converted into or divided into or developed into plumules and cotyledon. This one is divided or developed into plumule and cotyledon. At the same time, this portion is called as radicle. This is a complete what is the development of the mature embryo. Before the mature embryo, the double fertilization. After that double fertilization, you can tell it. Okay. After finishing the double fertilization, these are the changes occurs until the embryo formation takes place. Embryo develop into cotyledon, and that means root and shoot systems. These are the things. This is the way to grow. Okay. So any doubt in this area, Pa? Do you have
No, sir.
any doubt?
No, sir.
This is no. Okay. Go to the next one. See here, according to the cotyledon, they may be divided into two types. One is called as dicot embryo, Another one is called as monocot embryo. Dicot, that means cotyledon, how many is there? 2 is there, isn't it? Embryo. See that a typical dicot embryo consists of an embryonic axis and 2 cotyledon. That's why it is called as dicot. Okay, embryonic axis already we discussed, and followed by 2 cotyledon. The portion of the embryonic axis above the level of cotyledon is called as epicocyle. This is very, very important. Embryonal axis above the level of the cotyledon is called as epicotyles, which terminates with the plumule or stem tip. Okay, this is the very epicotyl, the upper part of the embryonal axis, otherwise called cotyledon, is called as epicotyle. How we can find out which one is epicotyl? When that dicot or a monocot growing season, in the primitive stage, when you are planting any one of the dry seeds in your house, what happened in the first initial stage? The first, what is that? The embryonic axis comes out. Okay, we can see it, epicocyle. Which one is epicocyle? Okay, the portion of the embryonal axis above the level of cotyledon is called as epicocyle, which terminate with the plumules or stem tips. This is plumules and stem tip is called as epicocyle. Okay. The portion of below the level of the cotyledon is hypococyle, that terminate and it is lower end in the radicle and roots. We can come to the conclusion. The center part is, what is that? The center part is embryonal axis. The above part of the embryonal axis, epicocyle. It produces what is that? Plumule and stem tips. The lower part is called as hypococyle, that produce radicle and root tips. Okay, but the root tip is covered with the root cap. This is also a very, very important. Okay, see that structures we can easily understand. So this is called as a, what is that? Dicot embryo. Inside the embryo, this one is called as plumules. These two are cotyledon. This is hypococyle. Hypococyle produce which one? Radicle and radicle. The radicle produce root covered with the root cap. So this one is what is that? Plumules. The upper part is called as hypococyle. This one is, sorry, hypococyle. This is epicocyle. Sorry, this one is called as epicocyle. This one is called hypococyle. In between, what will happen? Plumule present. Plumules produce which one? Suits them. The cotyledon provide nutrition for the plumules or embryo. So this is the structure of the dicot embryo. You note it down. This is also a very, very important thing. And monocot embryo. Okay, at this level we will finish it, I think. Okay, monocot embryo. The monocot, example, rice, maize, and etc. What about the dicot? Sisal aritinum. Okay, dicot, you can take an example. Anyone, so many things is there. Okay, mango, whatever it may be. Here, the monocot embryo, it consists of only one cotyledon called as, what is that? Scutellum. The embryonic axis has the radicle and the root cap enclosed by the sheath called as coleorhiza. Coleorhiza and the coleoptile. Okay. That means monocots is divided into 2 equal parts. The equal part division is called as scutellum. Below the scutellum, what happened? There are 2 types of parts is present in it. One is called as coleorhiza. Another one is called as coleoptile. Coleorhiza produces radicle. Coleoptile produce plumules. Okay, both are covered by a layer is called as, what is that? Coleoptile, coleorhiza. See that? Epicocyle, plumules. Hypococyle, what is that? Radicle. See the structure, you can easily understand. This one is called as, what is that? Monocot embryo. Okay, the diagram is not clear, but the structure given in your book like this. This 2 is called as coleoptile. Scutellum. This is the scutellum. Division of the embryo is like one, like biggest one is that division only, the lower part only they have given here. This one is called a scutellum. Below there is 2 part is that. Which one? Coleoptile. Okay, and also another one is called as coleorhiza. This is the cover. Okay, cover of radicle. Okay, this is the cover of radicle, and also root cap is also there present. So it's grow in the downward manner. bananas. So this is the way. It is protective. The root cap is used for the production of the production function, particularly the root. Okay, isn't it? Here the coleoptile is also there. Coleoptile, which one? The shoot apex. This one is called a shoot apex. It covers the which one? The coleoptile layer. This one is called as coleoptile layer. This one is called as coleorhiza layer. The coleorhiza layer protect the radicle, and coleoptile layer protect the shoot system. This is called as epiplast. It's additional what is that growth. This one is called as circuitulum. We will see the next slide. It will be better for your understanding. So this is already I show you. Okay, and this one is called as a dicot. This one is called as a monocot. Okay, see the diagram. You can easily understand. Okay, let's see the thing. Yeah, this one. See here, this is monocot seed, isn't it? So this is the endosperm. This portion is called a circuitulum. This portion only given in the elaborate manner in the before we saw that slide, So this is the elaborate portion. This is called a circuitulum. The monocot is divided into two parts. One is called as endosperm. This is the endosperm, the nutritive layer. Okay, but in this area circuitulum. See here, there is an upper portion. This is called as what a coleoptile, and below there is a plumule. This plumules produce a shoot system. The below part there is a radicle. The radicle covered by a layer is called as what is that? Coleorhiza. These are all are mostly present in the lower part of the seeds. This is monocot. Okay, see that dicot seed. This is the dicot seed. Cyser aritinum. See, this is the structure of cyser aritinum. Okay, this two are cotyledon. Already know about. Premier stage, what happened? This is the embryo. The embryonic axis is that. Okay, here the two part is that. Which one is the upper part is called as plumules. The plumule produces a shoot system. Otherwise called as cotyledon producer. The lower part is called as radicle. Radicle produces root system. Okay, these are the things. Okay. So this is the things. Today monocot embryo and a dicot embryo. How the double fertilization possible? So all those things we saw it, and post fertilization changes which occurs like that they told. So these things just I read very quickly. So just to understand it, after the fertilization finished, what happened? What are the changes which occur inside it? That is a very, very important things. Okay, post fertilization changes. Mature ovary become fruits. That means mature ovary only develop into the fruits. They may ask the question, match the four lines. Mature ovules become a seeds. Ovary wall, fruit wall, otherwise called as pericarp wall. This is already pericarp, epicarp, mesocarp, endocarp, fruit wall. So yeah, when we are going to study these things, we will study the fruits. Inducments is also seed coat. Seed coat, there are two types of seed coat is there. One is called as outer inductment and inner inductment. Outer inductment is nothing but the which one testa. Inner inductment, tegment. Okay, which one is the hardest here? Outer coast is the hardest one because the presence of stone cell. Why it is presence of stone cell? Stone cell otherwise called as scleroids. Scleroids examples. Which one By some sativum, isn't it? Okay. See here. So I will tell you this one. So which one is the very hardest coat is that? Take an examples of guava seeds also a hardest one because the outer coat is made up of which one scleroid cells. That is the patients. And then new cells completely degenerated. Sometimes it present in the form of perisperm. Already we saw that one is also extra. As a new cellus, sometimes it present in it. That extra new cellus is called as perisperm. Egg cell already but embryo. Egg cell is called as embryo. Embryo develop into what happened already. We saw it before 2 slides before, isn't it? Synergetic antipodal cell completely degenerated. Hilum of the ovule, hilum of the seeds, sac or seed. Funiculus of ovule, stalk of the seeds. This is very, very important. Funiculus. Okay, microphyll of the seeds. Chalasa of the ovule, chalasa of the seeds. Okay, these are the changes which occurs after the double fertilizations. Do you have any doubt in this area? So I think today we finished this area. Remaining things we will study that one. Angiosperm seeds, monocot seed and dicot seed. What are the difference between monocot seed and dicot seeds? All those things we will see the next classes. Okay. See, any doubt in this area? Ideas?
No, sir, no doubt.
No doubt. Can you explain fully? Syria. Okay, listen here. Completely already we discussed these things. You note it down. This one is a very, very important things. Aurum maculatum. This one polyploidy, recording that one. And also what are the types of endosperm? Endosperm, embryo, embryonic axis, how it will be formed, and double fertilization. And what are the types of endosperm? Polyploidy endosperm. And finally, what are the type of seeds? Monocot and dicot seeds that we discussed. After the fertilization, what are the changes occurs in the flowers? That things we will finish it. Okay. Thank you, my dears. Thank you for your cooperation. So just to go through the lesson, then only it will be better for your understanding. You have to attend more times. Okay, it will be better. Okay. Thank you. Thank you so much for your participations.
Thank you, sir.
Thank you.
Thank you,
Thank
sir.
Thank you. Thank you, ma'am. Thank you so much. Bye.